Welcome to G Fun Facts Online. Today, we're diving into a revolutionary battle plan against one of humanity's oldest foes, cancer. Forget what you think you know about germs, because in this story, the traditional villains become the heroes. Scientists are now forging an unlikely alliance, turning bacteria and viruses into a microscopic special forces unit designed to hunt and destroy tumors from the inside out. This groundbreaking strategy is called the Trojan Horse approach, and it's poised to change everything. The concept is as elegant as it is powerful. Use tumor-seeking bacteria to smuggle cancer-killing viruses deep inside a tumor, past the body's defenses, to unleash chaos in the heart of the enemy's fortress. Let's explore how this microbial alliance is creating a new era of living medicines. The idea of using microbes to fight cancer actually has surprisingly deep roots, stretching back more than a century. In the late 1800s, a New York surgeon named Dr. William B. Coley noticed something remarkable. One of his patients, who had an inoperable tumor in his neck, made a full recovery after contracting a severe bacterial skin infection. Intrigued, Coley developed what became known as Coley's toxins, a mixture of heat-killed bacteria that he injected into cancer patients. While his results were mixed and met with skepticism, he had planted a seed. He showed that provoking an immune response with bacteria could have an anti-cancer effect. This work eventually laid the foundation for modern bacterial immunotherapy, leading to treatments like the BCG vaccine, a weakened bacterium, which is now a standard of care for bladder cancer. At the same time, on a parallel track, another field was emerging: oncolytic virotherapy. Oncolytic literally means cancer-destroying. In the mid-20th century, researchers observed that some viral infections also seemed to cause tumors to shrink. They began experimenting with wild viruses, but faced two major problems. The patient's own immune system would often clear the virus before it could work, and there were serious safety concerns about using live, untamed viruses. For decades, these two fields, bacteriotherapy and virotherapy, progressed separately. But then, the revolution in genetic engineering changed the game. Scientists gained the ability to precisely edit the DNA of both bacteria and viruses. They could disarm them, make them safer, and even enhance their cancer-fighting abilities. This led to a pivotal moment in 2015, with the FDA approval of TVEC, a genetically modified herpes virus for treating advanced melanoma. With these new tools, researchers realized that the weaknesses of one approach could be covered by the strengths of the other, and so the Trojan Horse was conceived. The brilliance of this strategy lies in how it overcomes the body's own defenses. When you introduce a therapeutic virus into the bloodstream by itself, it's often a suicide mission. Many of us have pre-existing antibodies from past infections or vaccines that are trained to spot and neutralize these viruses on site. The virus is eliminated before it ever reaches the tumor. This is where the bacterial horse comes in. By placing the virus, or its genetic blueprint, inside a bacterium, it's hidden from the immune system. The bacterium acts as a living, mobile invisibility cloak, smuggling its deadly cargo through the bloodstream undetected. But the bacteria aren't just a delivery vehicle. They are expert navigators. Scientists choose specific types of bacteria for their natural ability to home in on tumors. Solid tumors create a very unique internal environment. As they grow rapidly, they outgrow their blood supply, creating zones with very little oxygen, a condition known as hypoxia. This hypoxic core, filled with nutrients from dying cancer cells, is a hostile wasteland for our own cells, but an absolute paradise for certain bacteria. One of the top recruits for this job is Salmonella, the same genus of bacteria known for causing food poisoning. Of course, these are attenuated, or weakened, strains. They are genetically disarmed so they can't make you sick, but they retain their natural instinct to seek out the low-oxygen, nutrient-rich conditions found deep inside tumors. Another key player is Clostridium. These are obligate anaerobes, meaning they can only grow where there is almost no oxygen. This makes them incredibly specific to tumors because they simply cannot survive in healthy, well-oxygenated tissue, providing a fantastic built-in safety feature. Once these bacterial vectors have infiltrated the tumor, the real mission begins, releasing the viral soldiers. The creation of these bacteriovirus hybrids is a masterclass in synthetic biology. First, the bacterial horse is meticulously engineered. As we mentioned, its disease-causing genes are deleted to make it safe. But crucially, it's also programmed with a self-destruct mechanism. Scientists install a lysis circuit in its genetic code. This circuit is often controlled by something called quorum sensing, which is how bacteria communicate and sense their own population density. They release signaling molecules, and when the concentration of these molecules hits a critical threshold, meaning enough bacteria have gathered inside the tumor, it triggers a gene that causes the bacteria to burst open, or lyse. This ensures the viral payload is released at the right time and in the right place. Next, the viral soldiers are armed. These oncolytic viruses are also genetically modified. Their ability to replicate is often restricted, so they can only multiply inside cancer cells, which have faulty antiviral defenses, leaving healthy cells alone. They can also be armed with extra genes. For example, a virus can be engineered to produce immune-stimulating proteins called cytokines. These proteins act like a battle cry, calling in the body's own immune cells to join the fight. So let's walk through the entire attack plan, step by step, using a real-world example called the CAPPSID system, developed at Columbia University. First, the engineered Salmonella bacteria carrying the genetic code for a cancer-killing virus are injected into the patient. The bacterial cell wall shields the virus from the immune system. Second, the bacteria travel through the bloodstream and, like tiny heat-seeking missiles, accumulate inside the tumor's hypoxic core. Third, once inside the tumor, the bacteria invade the cancer cells themselves. This is a key step. They get inside the target cells before deploying the weapon. Fourth, the quorum sensing circuit activates. The bacteria reach critical mass, and they all lyse, or burst, at once. This releases the viral genetic material directly into the cytoplasm of the cancer cells. Fifth, the cancer cell's own machinery is hijacked to start producing thousands of new virus particles. This viral army builds up until it bursts the cancer cell open, a process called oncolysis. This releases a new wave of viruses ready to infect and destroy neighboring cancer cells, creating a chain reaction. And here's the most important part. The sixth step is activating our own immune system. The explosive death of cancer cells caused by both the bacteria and the viruses releases a flood of tumor antigens, pieces of the cancer cells, and bacterial components. This combination acts as a massive danger signal, shocking the immune system out of its complacency. The tumor, which was previously hiding from the immune system, is now fully exposed. This triggers a powerful and targeted anti-tumor immune response, essentially training the body to recognize and kill cancer cells not just in the primary tumor, but potentially anywhere in the body. It's like setting off a bomb and a flare at the same time. The strategy has shown incredible promise in preclinical studies. The CAPPSID system, for instance, led to complete tumor regression in some mouse models, and critically, it worked even in mice that had pre-existing immunity to the virus, proving that the bacterial cloak is effective. Another study used engineered salmonella to deliver a different potent oncolytic virus, showing it could kill breast, pancreatic, and bone cancer cells. While most of this research is still in the lab, the results are overwhelmingly encouraging. The advantages are clear. This microbial alliance overcomes immune neutralization. It provides superior tumor targeting. It localizes the therapy right where it's needed. And it creates a self-amplifying cycle of cancer destruction that simultaneously recruits our own immune system to the fight. Of course, it's not without challenges. Using live, genetically modified microbes as medicine requires extreme safety precautions. Scientists are building in multiple safety switches and dependency circuits to ensure the bacteria and viruses can only function within the tumor. But the path from the lab to the clinic is long, and extensive human trials are needed to confirm the safety and efficacy of these complex living medicines. The Trojan Horse strategy represents a profound shift in how we think about fighting cancer. By harnessing the power of nature's own microscopic agents and refining them with cutting-edge synthetic biology, we are creating intelligent, adaptable, and living therapies. It's a fusion of microbiology and genetic engineering that is pushing the boundaries of medicine. The war on cancer is far from over, but with these tiny, powerful new allies on our side, the tide may finally be turning. Thanks for tuning in to G Fun Facts Online.